Hello everyone, welcome back to another class on regeneration biology. So in today's class, we will learn about extracellular matrix and its roles in tissue regeneration. So we have seen extracellular matrix and their importance in various regeneration aspects, and some emphasis has been given to the collagen and collagen 4 especially during the fin and the limb regeneration, which we discussed earlier. And today we will see an overall view on how the ECM is contributing, or extracellular matrix protein is contributing to the regeneration in a nutshell. So extracellular matrix mimicking biomaterials are effective tissue for the organ culture. They are tissue engineered scaffolds for regenerative medicine because of their biocompatibility, biodegradability, bioactivity, etc. So we will see some examples in the upcoming classes when we study the organ regeneration in vitro. But understand, the use of the biomaterials is exclusively dependent on how suitable it is to elicit a regenerative response or maintain a regenerative response which is similar to that provided by the extracellular matrix protein. So ECM mimicking biomaterials preserve natural microstructures and matrix related bioactive components, and they undergo continuous matrix remodeling upon transplantation. So many of these materials, when we are using, whether it's natural or artificial, once the cells start colonizing on them, then they start secreting their own matrix, and that will replace this artificial. Just like you would have heard about absorbable surgical sutures, which like internal suturing, when you do, you don't have to go to hospital and remove those suture. It will get absorbed into the body. So the interaction between host immune cells and the transplanted ECM mimicking biomaterials has attracted considerable attention in the regeneration biology field. Mainly, you have to choose a material which do not have too much of immunological implication. It do not attract. So these are all lessons you are learning from the natural ECM proteins. So the transplantation of biomaterials may initiate injuries and early pro-inflammation reactions characterized by the infiltration of the neutrophils and the M1 macrophages, which both of them are pro-inflammatory, can trigger a wound formation response. And the pro-inflammation reactions may lead to the degradation of the transplanted biomaterial and drive the matrix to a fetal-like state. Sometimes the initial pro-inflammation help in attracting more of the immune cells, and which in turn trigger a lot of the enzymatic protease enzymatic activity, which may help in clearing the ECM in a normal scenario, and clearing of these biomaterials because of their enzymatic activity in this artificially engineered scenario. So ECM degradation is a well-studied mechanism that is essential for regeneration, and this degradation also triggers lots of immunological response also, which is necessary in the initial phases of regeneration. So ECM degradation releases matrix-related bioactive components that act as signals for cell migration, cell proliferation, and cell differentiation. In late stages, the pro-inflammatory cells fade away, it is a necessity, and anti-inflammatory cells emerge, which involves macrophage polarization to M2 phenotype. Like we have discussed, M1 is pro-inflammatory, M2 is anti-inflammatory, and M2 phenotype, and also the leukocyte activation to helper 2 cells, that is Th2 or T helper cells, they comes into picture, which we can also call them as adaptive immune cells. So the anti-inflammatory cells interact with each other to facilitate matrix deposition and tissue reconstruction. The deposited ECM molecules serve as the vital components of the mature tissue and they can also influence the homeostasis. However, we should understand the dysregulation of the matrix remodeling can result in severe pathological response or pathological conditions such as aggressive inflammation and difficulty in healing, as it happens in the case of diabetic individual, and non-functional fibrosis. Fibrosis, we know, excessive deposition of the ECM proteins, and this is triggered mainly by the inflammatory response. Many times this M1 macrophages, when they are overly recruited into an injury spot, they will favor the inflammatory cytokine production, and as a result you will have excessive fibrosis that will lead to, and sometimes they can be benign, but sometimes it can even lead to debilitation of the organ function. So the characteristics of inflammatory response in matrix remodeling after transplantation of ECM, that is mimicking biomaterials, is very important or crucial in organ regeneration. This is what we should keep in our mind, how biocompatible the ECM material is, or the artificial material is, or how closely it is mimicking to that of the innate or the natural ECM, is very important for the success of or the establishment of the cells that is growing or deposited in that site of injury. A brief history of cardiac regeneration and its roles in the extracellular matrix. We know cardiac injury is one of the well-studied regeneration scenario in which the ischemia, the lack of blood supply, leads to formation of scar tissues, which can have serious implication in the contraction. So lots of studies are focused on that why can't we get, if the muscles can regenerate, skeletal muscles can regenerate, why not heart? And by tweaking that environment, where instead of the scar tissue, if you can get some muscular tissue, cardiac muscle tissue be replaced, then that will favor the overall pathology, or that will reduce the overall pathology or reduce the complexity because of this ischemia related pathology of the heart. So this is the overall idea. To push the field of cardiac regenerative medicine forward, several hurdles must overcome before we can develop therapeutic strategies to be implemented in a clinical setting. And every animal, whichever it gets a cardiac trouble or cardiac ischemia, they respond by regenerative response. Whether it is successful or not is the question. Firstly, we must understand why neonatal mammalian or zebrafish ECM, adult neonatal mammalian and adult zebrafish ECM, extracellular matrix, is able to induce adult cardiac regeneration. That is a very interesting question. But the adult mammalian heart cannot do this successfully. So to this end, advances in omics-based approaches, omics means it can be proteomics, RNAomics, genomics, etc. Omics-based approaches, including proteomics and RNA sequencing, will enable scientists to fully characterize the composition of the ECM and identify which components are no longer present in the adult myocardium. Because adult myocardium of the mammal doesn't regenerate. So what is so much change that occurred which do not allow them adult cardiomyocytes to regenerate? Next, we must determine how these candidate ECM components, individual candidate members of the ECM, are able to induce repair or regeneration of the injured myocardium. So this is another interesting question, in vitro and in vivo. So how they are able to induce a regenerative or reparative response. So that is very informative one should have, especially when you comparing the neonatal versus adult. Finally, the most crucial step will be the development of these ECM components into a synthetic hydrogel. We should bring in all the good stuff which is favoring a regeneration, introduce them into a synthetic hydrogel to facilitate and move into clinical application. So it's just like when you get a wound cut, you will apply a neosporin powder or a neosporin ointment, which has got the antibiotic, which will prevent the bacterial growth, and the healing has to happen naturally. Same way, can you put in the injured area something which has got this matrix components that will favor the regeneration? So this is the question. So these are all the approaches one has to take. Advances in the field of synthetic biomaterial have demonstrated that the potential for regeneration of such synthetic ECM hydrogels exists. It's a matter of how effectively we can inculcate or how effectively we can introduce this information into a in vivo condition or real animal condition. One recent study has demonstrated the potential of this by developing degradable elastin-like polypeptide hydrogel, which mimics that of native ECM. Which ECM? To that of the neonatal heart or neonatal heart which regenerate. So the mammalian heart has always been considered post-mitotic organ without the capacity of self-renewal. Just like your neurons, which is considered post-mitotic, means it is done and dusted with the mitosis event. It will not enter into the cell cycle. Where growth is achieved through cellular hypertrophy. It doesn't increase in number, but it increases in the size of a given cell. That is called hypertrophy. Recently, however, several studies have identified that the cardiomyocytes can proliferate to a limited extent in the postnatal heart of mice, not prenatal. Prenatal naturally it can, but not adult. Soon after birth, that is postnatal, they have the ability, they still retain the ability, although the heart development is completed, because baby heart versus adult heart size difference is there, but that size difference is because of hypertrophy. But postnatal heart of mice, rats and pigs and even humans, they have the ability to proliferate. Research has also shown that the immature mammalian heart possesses a greater regenerative potential than previously expected. Whatever we know a lot of regenerative potential in the postnatal heart. Embryonic, definitely it is there. And zebrafish throughout the adult stage, and even for other amphibian models also have the ability to regenerate the heart. Given the similarities between the adult zebrafish heart and the immature mammalian heart, the author Porollo et al., they induced cardiac damage by resectioning 50% of the ventricular apex in 1 day old neonatal mice. They have done. Surprisingly, what did they find? The apex was progressively regenerated, fully restoring the resected myocardium within 21 days, which is somewhat similar to that of adult zebrafish. So this indicates it has the potential, it has the ability. It is a question whether this quality can be retained in the adulthood of the mammal. So this regenerative ability of the immature heart was lost once the mice became over 7 days old. That means something goes wrong, at least a regeneration point of view, within 1 week of its birth. More recently, similar observations have been made in rats and pigs, not just restricted to that of mice. Even more fascinating is that several case reports have demonstrated a similar phenomenon in newborn human babies also. So, what we should know, within first few days after birth, the changes that occur in the animal tissue make them or render them less regenerative. This suggests that mammals retain surprising cardiac regenerative ability at birth, not the time point, at birth, which is lost upon maturation. Maturation doesn't mean that adulthood. Maturation means within few days of the organ maturation, or few days after born, which we refer to as organ maturation. Understanding which intrinsic mechanisms are involved in neonatal mammalian cardiac regeneration has triggered a new field of research into characterizing this innate ability for cardiac repair. Lots of people try to hunt for what is lost, what went wrong, what went missing within few days after birth, which may facilitate the identification of novel strategies for cardiac regeneration to fix a failing heart or failed heart. So we know ECM components, they can change after cardiac injury. We know their components change. This table shows that this is the members of the ECM, like collagen 1, collagen 3, collagen 4, 5, 6, fibrillin and fibronectin and hyaluronic acid. These are all part of the ECM component. Lots of proteins are there. And let us see regenerative situation, non-regenerative situation, and what can be done further. So regenerative situation are what? Adult zebrafish, neonatal rat, neonatal mice, and adult rat. Adult mice, they are non-regenerative. So regenerative are only 3, that is neonatal of rat and mice and zebrafish throughout. No neonatal both in neonatal as well as adult. They behave the same. If you see one ECM component called agrin studied in mice, it goes up. And remember, neonatal mice, it regenerates. You can see collagen 1 in zebrafish goes up, whereas in rat it goes down. That is a conundrum because neonatal rat regenerates, but the collagen 1 goes down. Now, collagen 3 in rat, neonatal rat, it goes down. Expression level goes down, but it regenerates. And in mice, it goes down. Even for collagen 1 in mice, it goes down, but it regenerates. And in zebrafish, collagen 4 goes up. And in rat, neonatal rat goes up. Mice, no reports are available. And in collagen 5 in zebrafish, it goes up. But neonatal rat, where it can regenerate, the collagen 5 goes down. So these some opposite results also seen, but a lot of them like a fibrillin, fibronectin in zebrafish goes up, rat it goes up, and even in mice it goes up. So, and when it comes to the adult non-regenerative scenario, that is the adult rat and adult mice, you can see a lot of them, like say collagen 1 goes down in both mice and rat, but it goes up in the case of adult rat, but it doesn't regenerate. So this table in general tells you what are the situations or what are the factors that is contributing to a regenerative as well as non-regenerative environment. So studies demonstrating the components involvement in cardiac regeneration. You can see recombinant agrin promotes cardiac regeneration. There is a research publication available. And fibronectin is important for zebrafish cardiac regeneration. There is a research study available. Hyaluronic acid inhibition blocks zebrafish cardiac regeneration. Research study available. Like hyaluronic acid, you can see in zebrafish it goes up, and in neonatal mice it goes up. Both situation regeneration happens, and in adult mice soon after cardiac injury the hyaluronic acid level goes down, no regeneration happens. So these are all things which should keep in your mind, that ECM components are having majority of them something similar among different species, which in a regenerating scenario like neonatal mammal and adult zebrafish, somewhat similar response is seen, and in non-regenerating scenario the levels are opposite. You can also see other components like laminin, periostin, and perlecan, and also thrombospodin, and also versican. So there are different ECM components are there. You can see their levels, and periostin involved in the neonatal but not adult cardiac regeneration, and there is published research study available. So we should keep in our mind that the ECM components are indeed regulated soon after injury, and how they are regulated, are they going up or are they going down, depends on the nature of that protein and also nature of the tissue, whether it's adult or neonatal, or whether they can be regenerative or non-regenerative. Accordingly, they have some common way of responding, and these are all the ways in which you can strategize. Can we think of some of these components introduced into a matrix protein, artificially put matrix protein, and we can facilitate a regenerative response. Say for example, if collagen 1 is downregulated in rat and it is downregulated in mice, and it is going up in the adult rat, which do not regenerate. So now when they are going down, can we think of having something introduced in that material that will downregulate the collagen 1 that can favor the regenerative response? So now lessons learned from the innate cardiac regeneration. We will learn, innate means the native regenerative ability, a brief history from across studied literature. Within the animal kingdom, there are numerous examples of spontaneous organ regeneration that is following injury. We have so many examples. In terms of cardiac regeneration, the zebrafish stands the most well studied and one of the well suited organism for studying the regeneration biology. Zebrafish can undergo complete cardiac regeneration without any scar being formed after resectioning up to 20% of the ventricular apex. It's a two-chambered heart and the ventricular apex is very much accessible and visible. That is the place people use it. And also understand the ventricular wall fibrillation is more problematic when it comes to cardiac dysfunctioning rather than the auricular wall, because ventricles supply the blood to the rest of the body. Complete regrowth of the amputated region, including the coronary vasculature and myocardium and endothelium, is achieved up to within around 60 days of post resection. 60 days means around 2 months of post damage. This results in a fully functional heart in the case of zebrafish. This model for cardiac regeneration has helped to unravel certain aspects of regenerative mechanism which we can hunt for in mammalian scenario, whether a given pathway is on or off in the case of a mammalian. Of course, neonatal mice or rat also comes into picture for these kind of analysis studies. Not that you directly compare into a non-regenerative model. Of course, non-regenerative model you can always compare to a regenerative model irrespective of the adult or non-adult situation. But what is most important is compare a well-studied pathway which has serious implication in regeneration, say for example in zebrafish, and look what is the status of that pathway in an adult. So this is the overall theme of research that is being carried out in the field. Still, since it is based on removing heart tissue, like normally you are cutting 20% of the ventricular apex by surgically heart tissue, rather than damage that naturally occurs, because someone gets a heart failure, no one is going to chip off his heart. There is a damage that is occurring due to ischemia. It is not the most accurate model to study human cardiac damage and repair. So to this end, recent study, one of the study has characterized cellular response and the ability of functional repair, that is regenerative capacity in general, of zebrafish heart after a cryo injury, where you take a small dry ice piece and you attach, which will stop the blood supply and oxygen supply, and that will lead to a similar to that of to produce that, unlike resection, unlike a surgical injury, more closely models pathophysiological processes undergone by human heart, especially in a ischemic condition. So damage was induced to 25% of the ventricle using cryocauterization. That is, you are giving a cryo injury or a low temperature injury, resulting in a massive cardiomyocyte cell death within the injured area and also near the coronary vasculature. So this is the overall idea of a cryo injury. Cell death, which occurs is because of this cryo injury, that is induced a proliferative response in the endocardium, epicardium and myocardium. 3 layers are there for the heart, ultimately resulting in the formation of a scar at the site of injury in a normal scenario. However, what we see, unlike in the injured human heart, the fibrotic scar tissue was degraded. First, a scar is formed, even in zebrafish, but the scar doesn't stay back. In humans and other mammals, adult mammals and adult humans, the scar stay back. That is the issue. The tissue was degraded and replaced by functional cardiac tissue in the case of zebrafish, suggesting that myocardial regeneration can occur even in the presence of a scar tissue. Means scar tissue is not debilitating the regenerative response. So if you have a scar tissue in a mammalian heart, scar tissue is not to be blamed, because in zebrafish also in cryo injury there is scar tissue formation which is removed. Although the damage of this tissue composition in terms of matrix and cellular components was not fully characterized, these results, what we see that the scar can be removed, indicate certain endogenous mechanisms are involved with scar tissue regression and cardiac tissue replacement. So we should understand it's not a given that if scar is there, it will be replaced. Scar is formed maybe a makeshift arrangement, and which is slowly replaced by a regenerative response. Or the blood shedding is not needed. You don't have to have an amputation. There is no need of a wound signaling for the cardiac regeneration. Characterizing these mechanisms underlying heart regeneration in such animals will offer way to identify novel strategies to overcome the limited regenerative response in mammals. So this is the overall idea. We should understand if a given animal's tissue can have scar formation, which is not a debilitating scenario. To start with, it is a debilitating scenario, but which can be overcome slowly. And why not such a strategy be adopted in a other animal, no matter whether it is a neonatal or adult. So here you can see an overview. It is being given a developmental ECM selection. How do you select? That is different sequencing work, proteomics or cardiac development, maturation, everything has to be looked into, and you take different stages, small heart and to a big heart. Into during developmental stage, you do sequencing proteomics and identify unique features, and in vitro cardiomyocytes proliferation can be studied very effectively, developmental of ECM components. And also you can go for strategy development. That means you can design a hydrogel that includes this unique developmental components that is identified. You can infuse into it, or you can even provide a recombinant ECM component into the system, so that will favor. And then comes the cardiac regeneration, that is in vivo cardiac regeneration can be introduced just either by delivering into the site that key components that is needed soon after an injury, and that is what is been mentioned. And we can also think of translation into clinical cardiac regenerative therapy, where you can get a cocktail, just like how you deliver a vaccine to a host to protect from infection. So advancement in the ECM based cardiac regenerative research, to advance the field of cardiac regeneration, it is essential to take the advantage of the innate regenerative ability of the fetal mammalian heart, which can regenerate effectively. So developmental ECM section will require first the utilization of advancement in the omics based approach, and such as RNA sequencing, proteomics, etc., to identify key ECM components, and that is essential for a developing heart. And then comes the developmental ECM components that will need to be screened in vitro for their capacity to influence cardiac regeneration. Just identifying doesn't make any sense unless it can do the similar job in an adult heart, which is a key component for which the induction of the cardiomyocytes proliferation must be obtained in the adult heart. Then only it is a useful thing. Otherwise, it is useless thing. Say, for example, if a French or a German person is able to read that manuscript in that language, doesn't mean that that is put in front of you. If you don't know that language, you will read as efficiently as the person. So knowledge is important. So tissue also, it varies. Embryonic heart is different from adult heart. So the delivery of the candidate developmental ECM components will have to be determined either by means of naturally produced ECM hydrogel, or by a synthetically produced ECM hydrogel, or by direct administration of these components in the recombinant form by injection into the matrix ECM of the injured or damaged heart. Having selected most suitable ECM component of cardiac regeneration and the appropriate delivery system, these will have to be tested for their in vivo capacity to induce cardiac regeneration. The first step will be to look around the animal's model or different model organisms such as adult mice or rat, either in a rodent model or in a large mammal model such as pig, for heart failure. Here, what we are learned so far is the development of an approach that will be implemented or administered in the animal after an injury to determine their efficiency in inducing cardiac regeneration and improving overall cardiac function. As the final step, the most crucial will be to translate this ECM based therapeutic strategy into clinical side. So what we have learned so far is there is potential, there is possibility, and because the ECM can hold the key in bringing in a regenerative scenario, because as you saw in the previous table, there are components that can mimic a regenerative environment, but getting the correct cocktail hold the key. So we should understand these angles or these approaches will enable a researcher to come up with most suited composition of the ECM that will allow regeneration of any tissue. We will learn more about regeneration biology in the next class. Thank you.